Hi, welcome to another episode of Tom Kennedy Science. I'm of course your host, Dr. Tom Kennedy. Now today we're going to start the first of a three-part series on the cell cycle. Now, you guys know the cell cycle. This is how the cell grows, replicates its DNA, divides, the daughter cells grow, replicate their DNA, and divide over and over and over again. And today what we're going to talk about is the importance of the cell cycle. I mean, why does this matter? And then I'm going to go over a general overview of the cell cycle. And then part two, I'm going to get into the nuts and bolts of mitosis. And then part three will be the control of the cell cycle, how you've got to get through all these different types of checkpoints. And to me, that's actually pretty fascinating. And in fact, the cell cycle itself is really fascinating. And when I start talking about mitosis, we get all these things about prophase, prometaphase. What kind of phase? Am I out of phase? Wait, I'm in phase. What's that phase all about? I got to name the phase based on the locations of the chromosomes. Why is this important? Well, I'm hoping that by the end of my series, you'll appreciate just how cool mitosis really is. And it's pretty awesome. But before I get started with all the nuts and bolts about how cells move chromosomes around, and then how you initiate or regulate or stop the cell cycle, you know me, you know where I'm going. I got to get the evolutionary perspective in here first, right? Because to me, everything in biology, of course, goes back to evolution. And I kind of like that, I like talking about evolution. So let's go back in time, way back in time. Let's go back in time about 3.8 billion years ago. It's the dawn of life. And we got our first cells. And our first cells, of course, emerged from abiotic processes. It's called abiogenesis. And they emerged, from these geological processes. Now, what we don't know is that if life actually evolved more than once and then was just wiped out. But we do know that in today's world, all the life we see is a direct descendant of those ancient ancestors. So the cell cycle, this cycle of growth, reproduction is very important. Now, most biology, in fact, to be living, they'll say, hey, you got to reproduce. Well, not so fast. Let's think about it this way. To be living, life is, okay, I'm going to say complicated here. Life is a result of emergent properties of a complex system that's using energy to create order. So that's why I say life may have popped up multiple times. However, all those early experiments in life, we have no idea if they were there or not, didn't make it. But we know that all life came from this original population of cells that learned how to reproduce. So what this means is that reproduction is important for two reasons. One, it ensures the continuity of life, right? Ever since life emerged, at least the life that led to us, about 4 billion years ago, it reproduced, and that ensured that life would continue on. If life never evolved the ability to reproduce, what would happen is you'd get some life, or proto-life, if you want to say it can't reproduce, it's not really alive, some type of proto-life would arise and then just get wiped out. Now what that means is something really cool. You and me, and every life you see on this planet, but you are the direct descendant of a continuous lineage dating back 3.8 billion years to the dawn of life. Now that's pretty awesome, isn't it? Think about that. You're the direct descendant of these ancient ancestors. And that to me is really cool. Now reproduction is also important for one other thing. We would not be living in a world teeming with life if it weren't for reproduction, because that ensured the continuity of life. It ensured that life could continue on. But it's also important for one other reason too. When you think about reproduction is basically I'm copying the information I need to have a living organism, right? We have this DNA, this the set of codes. It's like a cookbook for like the instructions to have a living organism. But every time you copy something down, have you ever copied a mistake? Oh yeah. With every single generation, there's mistakes. We call those mistakes mutations. And those mutations create variation within a population. And it is natural selection, the driving force of evolution that operates on those changes. And as a result of reproduction, leading to mutations, we have a world not just teeming with life, but we have a world that is teeming with millions of different types of species. So that's why reproduction is important from a very broad point of view, right? It ensures a continuity of life, of which you are this continuous process. And it also ensures that there's variation, that evolution can act on life so that life can diversify to do so many different and amazing things. It's pretty awesome. I just love thinking about like this origins of life. And of course, I said we're the direct descendants. It gets a little complicated here, right? The very first living organisms were simple. They were prokaryotic cells. They were small. They were structurally simple, and they included the archaeans and the bacteria. In fact, some call them eubacteria or archaeobacteria. But those two lineages diverge almost at the start of life, and there's reasons why we believe that. And of course, they evolved, but they were kind of trapped in their size for a long time until, of course, endosymbiosis came along, and you've got the merger of an archaean with a bacteria, and of course, that led to eukaryotic cells. Eukaryotic cells were much more efficient at extracting energy from the environment. Thank you, mitochondria. And as a result, life evolved into multicellular organisms as well. So that's pretty awesome, isn't it? Now, there's this thing in biology. It's been around for a while, over 100 years, and it's called cell theory. Now, cell theory has two parts. The first part, it says all cells come from pre-existing cells, and that cells are the basic unit of life. And that's true for today's world. The cells are the basic unit of life. They're the smallest functional thing that is got all these processes that make it living. And in today's world, there's no abiogenesis taking place, or I would be incredibly shocked if abiogenesis is taking place because life is everywhere. So ever since life got started, because we share these features of all of our common ancestors, all cells have come from pre-existing cells since abiogenesis, which is kind of cool. So there you have it. We're this unbroken lineage. And of course, it's a little complicated because of endosymbiosis, right? So, that was a merger of two organisms that led to eukaryotes, which is just fascinating. So prokaryotes, they reproduce asexually. And I'm going to talk more about that later. And then eukaryotes, they evolved a new type of reproduction to go along with asexual reproduction. It's sexual reproduction, and that's meiosis. And we're not going to talk about that here. But mitosis is creating genetically identical, more or less offspring. The more or less part comes from the number of mutations. So like I said, every single time a cell replicates itself, there's at least one or two mutations, if not hundreds. And that's why the daughter cells are never exactly the same as the parent cells, even though they're just copying the DNA. And then of course, sexual reproduction is you're taking two genomes and you're combining them into a single organism. And of course, that's how most animals reproduce. So, but what we're going to focus on here is, of course, asexual reproduction. Now, this is, of course, incredibly important because eukaryotes can reproduce both sexually and asexually. Unicellular organisms, they can, if the times are good, they can reproduce sexually. If they need to, they can also increase their numbers rapidly through a process called mitosis. Now, for you and I, in animals, we are multicellular organisms, and we rely not just on sexual reproduction to create a new organism, but we are multicellular, made up of 17 to 20 trillion cells. That's so many. So we rely on asexual reproduction, mitosis, for our growth and development. I mean, every single one of us started out as a zygote, a single-celled organism, and through mitosis, we grow into an adult. And so that's our growth. That's our development. It's also for maintaining homeostasis. Think about this. Every day you are alive, you lose 50 to 70 billion cells. That's with a B. That's a lot of cells. And those cells are lost. So you're constantly having mitosis replace all of your epithelial cells. Those are the cells of the lining of your body, like think skin cells. And of course, to repair tissue damage. And if some of you have been following me along this semester, last week I fell and nailed myself, and I'll have some pictures to show you how that wound is slowly starting to heal up. It's really itchy right now. And but it's repairing that tissue damage, and those skin cells are repairing themselves using mitosis. I always like to make sure that we have a little foundational knowledge. So in cell biology, we don't really get into genetics too much. But when you start talking about chromosomes and cell division, it's good to know a few of the words out there. So the genome, right? This is like all of your genes, all 3+ billion base pairs, and the code for over 20 something thousand genes. And a gene can code for a protein or it can code for some type of RNA. Chromosome. Some means body. Chromo means colored. About 130 years ago, 140 years ago, scientists were able to like dye these cells and they saw these colored bodies and they'd move around when the cell was dividing. They didn't know what they were, so they called them chromosomes. And you can only see a chromosome during mitosis. During all other times of the cells, when they're not dividing, that genetic material that has all your DNA, it's in a spread out decondensed form called the chromatin. All right, and that's a bunch of DNA and proteins that are there to like service the DNA and do transcription, copying the information of DNA to RNA so you can make proteins. And then when you replicate your chromosomes, you have these things called sister chromatids. And then lastly, your somatic cells, soma, there it is again. That means body. Those are the cells of your body. So when you look at me, you're seeing my somatic cells. And then of course, the gametes include the sperm and egg. So let's take a very close look here. What you're seeing is a single chromosome. In the electron microscope, the picture, that's a single chromosome. Now, it's replicated. So there's two copies of it right there. But because it's held together by a structure called a centromere, we were going to call that one chromosome. And it's during mitosis that we'll actually split that apart and it will become two different chromosomes. So that's important to understand there. Okay, so there it is. You can see the centromere. This is a very important term. Centromere is a region of your DNA that holds the sister chromatids together prior to the cell coming apart. Okay, here's some other words. The somatic cells. These are the cells of our body. Skin cells, liver cells, muscle cells. Basically every cell in your body is a somatic cell. They are diploid. Di means two. Ploidy has to do with the number of chromosomes. So if you're diploid, you got two copies of your chromosome. You got 23 from your mom. You got 23 from your dad. And so now you have 46 total chromosomes, but you've got a duplicate set, right? You got two copies of your genome. Now, on each of those, what we call homologous chromosomes, you have genes located at specific locations, okay? Now, you might have different versions of those genes. You might get A blood from your mom and B blood from your dad. Those would be alleles, all right? So that helps out. And then the picture on your right, that is the karyotype. And a karyotype is just an image of all of our chromosomes. Notice chromosomes 1 through 22, you've got two of them. One set from mom, one set from dad. And like I said, they're called homologous chromosomes because they carry the same genes. And then, of course, the 23rd pair are our sex chromosomes, which are the X and the Y. And we label our chromosomes in numbers from 1 to 22. And if you look, you can pretty much figure out how they did this. Number 1 is your largest chromosome, and 22 is your smallest chromosome. So, changes in chromosomes. Like I said, when the cell is not dividing, when it's not in mitosis, it's what we call interphase. And we'll come to that in a second. And it's in this form called chromatin. You really can't see it except with an electron microscope. But our DNA is chopped up into 23 of these chromosomes that are linear. And if you look at the picture, you can see that the ends are not connected. That's why they're called linear. In bacteria, the ends are connected, so it's circular. And when you get to genetics, you're going to find out that that's actually quite an important point, that we have linear DNA, because it limits the number of replications that our cells can do unless something goes wrong. It's a defense against cancer. So here it is. Here is our cell cycle. We alternate between the M phase, which is known as the mitotic phase. That's when the cell is actually dividing. And then you have what is called interphase. And that's when the cell is growing, preparing for the mitotic phase, and also replicating as DNA. So, mitotic phase, cell division. It's split into cytokinesis. Cyto means cell. Kinesis means kinesing means to move. So in cytokinesis, that's when we split the cytoplasm in half and all the organelles. And then mitosis, this is the actual division of the nuclear material. So if you ever heard prophase, prometaphase, metaphase, anaphase, yeah, that's what's happening on mitosis. Now interphase, if you notice, there's G1, S, G2. G1 stands for first gap. S is DNA synthesis, S is for synthesis, and G2 is a second gap. And these gaps, they get their name because when you come out of mitosis and the cell is small, right? There's a gap between the time that the cell ends cell division and when it replicates its DNA. And then there's a second gap between the time you replicate your DNA and go into mitosis, at least most of the time. There are some exceptions to that as well. Now, what we're going to talk about in another lecture is all of the different controls of the cell cycle. A cell does not just simply go round and round and round and round through these cell cycles. You must have chemical signals. That is important. You get growth factors. That's why your skin maintains itself the same way all the time. And then when you get a cut, then you'll send out these growth factors and those cells will start to go through the cell cycle. But even if you get a growth factor, remember from chemical signaling, you still have to pass what is called a checkpoint. The cell has the ability to check for damage to the DNA and to the chromosomes and then attempt to repair it. It also will check to make sure the chromosomes are pulled apart correctly during mitosis. It will also make sure that chromosomes are lined up in mitosis. It also makes sure DNA has been replicated, and it will also make sure the cell has enough energy to go through mitosis. So like I said, these checkpoints are really, really, really important to go through. So just kind of recapping really quick, you got your first gap, right? That's you come right out of mitosis and you're about half the size of your parent cells. It makes sense, right? You took your cell and you made it in two. And those cells are going to grow. They're going to add new organelles. It's also going to start making sure all the DNA is repaired. Now, if this cell gets a cell signal, it will then proceed to S phase where it will replicate its DNA, provided it passes through the G1 checkpoint. And then once it replicates its DNA, it transitions into the G2 or the second gap. It'll undergo a lot of changes at the cellular level, preparing for the mitotic phase. And it will also, once again, it might grow. It might repair the DNA. And then if it can't fix it, or if there's damage to the DNA, or if the cell doesn't have enough size or enough energy, then the cell can exit the cell cycle. And in fact, lots of cells exit the cell cycle. It's called the G0. I've heard it called quiescence phase. I wouldn't really call it a resting phase because your muscles, your nerve cells, they might spend their entire life in the G0 phase. They're out of the cell cycle. They're not growing and dividing, but they're totally active. I mean, think about your neurons. They're in your muscles. They're constantly working throughout your life. So I wouldn't call that a resting, but I would call it a non-growing or non-dividing stage. So for some cells, that's absolutely normal. For damaged cells, they might go out of the cell cycle, build up their reserves of enough energy to complete the cell cycle, or they might try to fix problems with their DNA. And then there's another way that cells can exit the cell cycle. Like I said, some of them, that's just what a mature cell does. A neuron just exits the cell cycle because it doesn't need to grow anymore. And then, of course, as I said, you've got 50 to 70 billion cells that kill themselves every day. And you can exit the cell cycle through apoptosis, which is programmed cell death. And I talked a lot about this in the previous chapter in my previous series on chemical signaling, but this is one way that cells can exit the cell cycle, and they can do this either internally if they sense damage, or they can get it through a cell signal. So you can get the death signal and these cells will commit suicide. Importantly, this is different from necrosis. Necrosis is where the cell becomes damaged and just explodes all over the place, spilling its contents, and that can lead to inflammation and other problems like gangrene. So apoptosis is actually a good thing because it's an orderly process of letting these cells die so that we can maintain, fresh cells on the linings of our body. And of course, we need apoptosis for early on in development. Remember the webbing? You'd have webbing between our fingers if it weren't for apoptosis. Why would we have webbing between our fingers? Because we evolved from fish? No, I'm curious. I'm not kidding. We evolved from fish like 375 million years ago, called a lobed fin fish. So it kind of makes sense to me that we would have webbing between our fingers when you think that our limbs are actually modified fish fins. How cool is that? And of course, cells can also exit the cell cycle if they become infected by viruses, if they become cancerous, or if they become damaged in some other ways through some type of internal cellular stress. A lot of times it could be too much oxidative damage. And then in that case, like if the mitochondria start having problems, if they lose their membrane potential, they'll initiate apoptosis. And once you start, you're done. You're going down it. So the nice thing about it is apoptosis does not spill its contents everywhere. It creates these small little bodies called blebs that are picked up by your white blood cells that can be easily disposed of. And the other thing that apoptosis does is it releases these things called nucleases and DNA aces. So it's chopping up all your nucleotides and proteins as well, which is important. Okay, and so this is very orderly here. And of course, apoptosis is very important because cancer occurs in damaged cells. And cancer cells escape the normal controls and checkpoints of our cell cycle. They bypass those checkpoints, they replicate with damaged DNA, and they have evaded apoptosis. And so a lot of people have defined cancer as a failure of apoptosis. And in that case, instead of committing cell death if they're damaged, they continue to replicate over and over and over again. And there's more to cancer than that, because cancer cells also turn on an enzyme called telomerase, which extends our telomeres. Remember I told you that our DNA is linear? You have a limited number of replications, but we're not born at age whatever, 45, however old your parents is. No, you're born at age 0 because you've got an enzyme called telomerase that will reset the clock each generation that you reproduce. So it's active in your gametes, not active in your somatic cells, unless you're cancerous. And cancer, one of the things about cancer is that it can knock out those control regions. Telomerase becomes active again, and these cells can be basically immortal. Okay, so that is my brief summary of the cell cycle. And my next episode is going to be on mitosis. We're going to go through the actual steps of mitosis next. All right, this has been another episode of Tom Kennedy Science.